going to look at the Cranial Vault. These are things that we're going to talk about today, and you'll see again in the lab. As far as the lab goes this week, I'll be in there pretty much working with pro sectors, but if you want to come in for some extra work on the skulls, we're going to probably help you, and probably be in there from about 8:30 to 10:30. Okay? Saturday, I'm sorry. Yeah, this evening from 8:30 to 10:30.
Wait, this Saturday.
Oh, come on. This Saturday. What day is today's Monday? All right, so I'm going to look at a lot of structures. We've got these linings in the cranial vault that we've been talking about shortly, just by definition. I mean, we've talked a little bit about them. Yes?
Is there some place where you posted a list of every term we need to have memorized for the lab final?
Gray's and I mean, you want a list of every absolute term?
Ideally,
I mean,
yes.
then that's basically a textbook. Okay? The two textbooks that I would recommend for you are Anatomy for Dentistry, okay, which is by Eric Baker. Pretty much everything is in there. And also, there is a book by Hyatt, which is basically called Head and Neck Anatomy, that you can use as well. I mean, if you want a list of terms, I'll be giving you a list of terms as we go back into the lab.
Yeah, because with heart and lung, then we could just go through and check it off if you found them.
In the lab, right?
Yeah, right.
Okay. Yeah, for this, so because there'll be a lab on this, and there'll be a checklist off for this, okay? Before that, we're going to have a lab on superficial income face, and there'll be a checklist off for that.
Yeah.
Okay.
Where does one find those checklists?
I'll post them on
Okay.
Canvas, okay? I'll post them on Canvas. But basically, as I said before, okay, you folks are the clinicians of the head and neck, okay? More so than you can appreciate at this point in time, but you should know everything. Okay, there shouldn't be any stone unturned, okay, so to speak. All right, so let's look inside here. We're going to get in there by doing some kind of top to bottom type dissections, but we have these meningeal tissues. Meningeal tissues are supporting tissues. There's basically, we'll see 3 layers. They're a very tough layer called dura mater, a layer that is sometimes like a spider web called arachnoid mater, and a very delicate thin layer that lies over the surface of the brain, which is basically a very soft layer called pia mater. Actually, you know, as far as the term, talking about terms, M-A-T-T-E-R is matter, okay? But when we say dura mater, M-A-T-E-R, or either dura arachnoid or pia mater, the term actually defines as mother. So, you know, somewhat sarcastically or facetiously, dura mater is tough mother, okay? Arachnoid mater, I just call it that spider web, okay? And then pia mater is soft mother, okay? So as compared to when we're talking about matter in general, you know, things that make up all kinds of, whether it's tissues in the human body, bones, et cetera, and things like that. So there are 2 layers that give rise to these 3 layers. The 2 developmental regions, or mesenchymal regions that will differentiate. One is called pachymeninges, and that gives rise to the dura mater. And the dura mater is an external fibrous layer that arises from mesenchymal tissue. Leptomeninges gives rise to the inner 2 layers, which are arachnoid and pia. We'll see those few layers, it's just a set. They also arise from mesenchymal, but the mesenchyme interacts with the breast specifically. Okay, pachymeninges actually comes in relationship from what we call neurotubular epithelium, okay, or the central nervous system line. Okay. So if we, I think we've looked at this before, we're looking at the scalp. When we look at this layering here, from outside in, this uppermost layer that's part of the superficial head is your scalp. Okay, we talked a little bit about that in the laboratory because your scalp is a living tissue, just like every tissue in here is a living tissue. So in order to be a living tissue, it has to have at least two things. It has to have an arterial supply and a venous return. Okay. Interestingly enough, this tissue, since it's on the outside of the cranial bones, these bones here, frontal bone, parietal bones, to the top of the occipital bone, okay, a couple of bones and so forth, venous return can either go external and back into a circulation referred to as the external jugular vein, okay, which we'll talk about a little bit today. But also it can go internal from the outside of the skull into the inside of the skull. So those of you who were here on Saturday afternoon, what do we say those types of openings are called? Emissary, okay? So those are called emissary openings because they are basically taking a structure, in this case a vessel, from one surface to another. And in this case, the majority of emissary veins take venous channels from outside to inside or inside to outside. And the reason I'd say, it's kind of a vice versa type of relationship is that venous channels in the head don't have valves, so blood can flow in either direction based upon gravitational forces. Okay, and that's an important thing to understand. So, for example, as we look at this, we'll look at, we talked about these diploic venous channels and so forth. The scalp will drain into diploic venous channels between the inner dense bone and the outer dense bone. And those channels, those diploic channels, can then drain internally into the cranial vault, in particular in relationship with the dura mater, but also they can interconnect with dural venous channels we'll talk about this afternoon that are associated with venous drainage off the surface of the brain. So you can get some kind of, it sounds, it doesn't, when you think about it, it's like, well, how could that happen? How could we get a intracranial infection from a scalp infection? But we can. Just like we can get an intracranial infection from a tooth abscess, a dental abscess, or some type of sinusitis, or some type of facial infection, okay, associated with a facial vein and its drainage. Okay, so here, we start out here over on your right, and we have the skin. And then underneath the skin, this is the front of the scalp, and this is the back. And as we go from right to left, we have, these things got misplaced. All right, so let's redraw them. That's cute. I just does that to confuse you. That's my rule. Skin, epicranial aponeurosis. Okay, and another name for this is the gala aponeurotica. And then And then the next layer in is periosteum. And then under the periosteum is bone. And then once we go through bone, we get to the outside of this layer, which is duramater. This almost came out correct here. And then we have this covering blood vessels on the surface of the brain, that's the arachnoid mater. And then we have this innermost layer associated with covering neuron populations, okay? We look at the spinal cord, we know we have this gray matter core, okay? So what's the difference between gray matter and white matter? What's happening in gray matter? What kind of tissue do we have in gray matter?
Somas?
So what kind of cells? Neuron, right, we have nerve cell bodies, okay? And that's surrounded by a shell. Sometimes we use the term cortical to talk about a shell-like entity. So this shell is white matter, and what's happening there, right?
The process is axons.
Okay. Well, axons, tracts, fiber tracts, can be ascending from the periphery to the brain and from the brain down to the periphery, are surrounding that. Now, as they ascend, these fiber tracts eventually are going to get to neurons at higher centers. And so some of those neuron populations we would find in association with nuclei. Talk more about these brainstem nuclei when we get into neuro next year first semester. But things like the reticular formation, okay? Things like the medullary nuclear populations, like nucleus cuneatus, nucleus gracilis, things like that. Raphe nuclei, okay? The thalamus, things like that, which is really diencephalic more than it is brainstem. And then cerebellum, or higher up, highest up into the neuron populations of the cerebral cortex, okay? Which is, kind of makes us who we are, both as an organism, but as a living, breathing, thinking organism. Okay? So the neuron populations in this highest center are on the outside. They're on the outermost side, just underneath these tissue layers, just deep to the cranial bone, deep to the skull. That's why it's there. That's why it's there, to protect these neuron populations. And we'll talk about, there's a couple ways that it does it, not just because of the thickness of the bone, but also because of some other substances that are in here, for example, cerebrospinal fluid, which actually acts like what we would call a hydrostatic cushion between the outside of the bone and these neuron populations. Okay, and then information coming down comes from these neurons, and that is efferent information, and that is going out to supply some type of an effector organ. That effector organ can be skeletal muscle somewhere in the body. That effector organ can be smooth muscle, or glandular smooth muscle, or cardiac muscle, or just nuclear populations sitting in the brainstem itself in relationship to these cranial nerves from an efferent perspective. Okay, so these layers are protecting it. Now, what we see here, here's the front, here's the back. This is a little slip of skeletal muscle here. This is a little slip of skeletal muscle back here. And then in between them is this aponeurosis, this galaponeurotica, or epicranial aponeurosis, which interconnects the two. Okay, and that those two muscles actually have a relationship both on kind of folding or corrugating the muscles in the back or the tissue in the back of your scalp, along with the tissues in the front of your scalp. Okay, and once again, these muscles sometimes are associated with facial expression or some type of body language that we might display in relationship to how we're feeling. Okay, so it's kind of interesting. The anatomy that the American Plains Indians had was that they knew that if they took a sharp blade and went through the scalp all the way around, because the galaponeurotica was covering the entire scalp, it would be very easy to remove. Okay? It was just a very simple dissection procedure. Okay? So, if you cut through here, we'll see when we go into the lab, it just peels right back off the top of the scalp, off the top of the skull. All right, so skin, gallop neurotica. This layer here is periosteum. The role of periosteum is to try and maintain some type of a transport tissue to allow blood flow to go into bone where red blood cells are being produced, okay? And those red blood cells coming back into circulation, okay, as erythrocytes, as opposed to just forming as erythroblasts within bone, okay? And then we have the bone itself. This is dense bone, very dense bone, so we call it cortical. We call bone that's not dense or more spongy, we call that cancellous, okay? So this is dense cortical bone. And then we go one layer deep to the bone, and we have this tissue, and this is dura mater. Now, dura mater is said to be a bilayer. It has an outside that faces the inside of the skull bone, okay, which we call periosteal dura, okay? And we have a side that is very smooth and shiny that faces the brain, and we call that endosteal dura, okay? So what we're seeing here is a periosteal dura that would be against the bone. Under this would be the endosteal dura, and trapped between those two layers is vascular bed. And this vascular bed, this is a highly vascularized arterial bed that is referred to as meningeal vessels, meningeal arteries, and they come from two places. They either come from a branch of the deep face, referred to, you had Dr. Wolf talking about the deep face, right? Okay, didn't she talk about the branches of the maxillary artery? Okay, so what branch of the maxillary artery comes here? Just gave you sort of a hint. Middle meningeal, okay? That's a branch of the maxillary artery. And then in the back, there is a branch that comes, we talked a little bit about this in the lab on Saturday. There's a branch that's believed to come from the occipital artery that can either enter through the foramen magnum, as well as some of these other structures we talked about, or it can actually enter through the jugular foramen, okay? And that's called the posterior meningeal artery, okay? So those two arteries basically are what supply the inner and outer surfaces of the dura mater. The periosteal portion we'll actually see on the slide as we go in the endosteal portion, okay? Then under here is this reason it's called the arachnoid, is that between the outside of the arachnoid, or the arachnoid mater's more external portion, and its internal portion, which it actually attaches to the pia mater by like little tentacular type of, or trabecular type of tissue extensions, okay? So it looked like a spider web. So what are spiders? Spiders are arachnids. So we came up with this term, arachnoid mater, okay? Now, between the pia mater and the arachnoid mater, there's a space that's called the subarachnoid space that will be filled with a extra, ultrafiltrate of blood, just like the kidneys ultrafilters blood. The brain also has a mechanism that we'll see called choroid plexus, which is an ultrafilter of blood that goes to the brain, and it creates this ultrafiltrated blood called cerebrospinal fluid. Okay. The pia mater plays a role in the ability, in allowing certain substances to pass to the neurons, but other substances not. So this is an area we'll talk about more in neuro called blood-brain barrier, okay? Where the brain is very selective in relationship to what it allows to pass in close proximity to these neuron populations. Okay. So we're not going to talk too much about blood-brain barrier now, but we will next. So on the surface of the brain are arterial vessels that are branching from deep to superficial, and there are venous channels which are branching from superficial to deep. Okay, and we'll look at some of these this afternoon. So it kind of covers this, and then if you go one layer deep, you can't even see pia mater because it's really only one cell layer thick. Okay, so it's very, very thin. You know, one cell layer is tough to see without, let's say, even a light microscope. But they're so close together that most authors, when they talk about them, they talk about them as a pair of tissues called pia arachnoid. Called pia arachnoid. So that's as we go from outside. In between the cortical layers of bone, as we're going from the outer surface to the inner surface, in here is this space we call the diploic space, or just more simply, the diplo. German term of the nuance over the E. And these are deep venous channels, yes.
On the last side, the vascularized arterial bed, that's between the two layers of the dura mater on the last slide?
Yes.
Okay.
The meningeal arteries. We're not there yet.
The one
We're
that you
still,
mentioned in the last
we're
slide.
still between the outer bone, cortical bone, and the inner cortical bone. We'll get there. I'll show you, we'll see that actually in a schematic, not in a schematic, actually in a photo micro. So these are between the layers. So once we get under the bone, we haven't gotten into the inside of the skull yet. There is this bone that is spongy bone, or cancellous bone, and in there are these diploic venous channels. And this is how some type of vector could enter into, from the outside into the inside of the skull. And so if we look at this from a schematic perspective, we have here, source, but here's the dura mater, and in the dura mater, here's the periosteal dura facing the skull, and the endosteal dura facing really the brain down here. Okay, we've got blood vessels. We've got arterial blood vessels, and then we've got these dense fibrous connective tissue cells that are pretty much dense collagen, which are sitting in here, and that is the packing meninges. Okay, that is the packing meninges. There's kind of a loose network deep to the endosteal dura that interrelates with this cells here. This is the arachnoid cells. The arachnoid trabeculae coming down. This, I don't know how this happens, but it happens sometimes. I'll have to reset it after this and get it reposted for you. But these are these trabeculae. Okay? So there are also vessels in here. Some of these vessels in this subarachnoid space now are arteries, branches of that large internal carotid artery that came in here from the outside of the skull and worked its way through the petrosal bone and then back behind the orbit and then up to the surface of the brain. So there are also venous channels on the surface of the brain that are covered by this arachnoid mater. And then down here, this is the pia mater. So we would call this layer, all this layer here and here, we would call it as a milieu, we would call it pia arachnoid tissue. Okay? Now, one of the other things that happens during development is that these venous channels somehow have to enter into channels that allow the venous return to go back into circulation. Okay? The primary, once again, the venous channels are avalvular, so they can actually, by gravitational forces, go forward or go backward. But the primary drainage through this cavity that we'll see is the internal jugular vein. Okay? And there are channels that are formed. I showed you an occipital bone in the lab, those of you that had deep grooves in what's sometimes called the squamous or the flat portion of the occipital bone. And what do we call those channels that made those indentations? dural venous sinuses, okay? There you go. If you don't know it now, know it before the first formative exam. But the dura mater is basically a bag that surrounds the brain. Here now we're looking, this is the periosteal dura side of the dura mater. On the top of it there is a channel that runs down the middle that is basically formed by dura mater, okay? It is not a true venous channel from the standpoint of its histology, but it is a venous transport channel for moving blood on the top of the cerebral cortex and also deep within the cerebral cortex, we'll see, back into circulation. So, let's see. I think they tried to show you. Kind of see how granular that is over here? What do we call these little granular entities that cause these depressions in the underside of the gallery? Surprise! Arachnoid granulations. Okay? And the arachnoid granulations are one-way valves from the subarachnoid space back into, in this case, the superior sagittal sinus. Okay? So this is the superior sagittal sinus here. The arrow is staying close enough to the pia arachnoid, but this here is the superior sagittal sinus. Okay, this, what we're looking at out here, this is periosteal dura. This is really tough to get that. I mean, when they get something like this, is what I like about McMinn. They really worked hard to get some of the dissections they had, and it's really good in the head and neck. We have a graduate student at the podiatry school that's working with us from Kent, and he was able to get this out as a whole dural bag containing the brain this past year, which is kind of tough. We should have paid him more for that. Okay, but we didn't. He's basically a graduate slave. But if we now go to the inside here, we can see these blood vessels. Okay, so now sitting on this layer here, we've kind of shaved back the periosteal dura, and we're looking at it lying on the outside of the endosteal dura. This is the middle meningeal artery. Here's the posterior meningeal artery here. The middle meningeal artery will supply 2/3 of the cranial vault dura mater. Okay, the anterior cranial fossa and the middle cranial fossa will overlap a little bit with the posterior meningeal with the posterior cranial fossa. Okay, so this is the middle. This is the middle meningeal artery. This is the posterior meningeal artery.
And the middle supplies 2/3 of what?
2/3 of the cranial vault dura. Frontal fossa, the anterior fossa, and the middle fossa. Okay, middle cranial fossa, anterior cranial fossa. See this right here? Any of you ever read the Bible story about David and Goliath? Okay. So David had to be a real good shot, because what he did is that he hit Goliath right here, okay, with this large rock, and caused a fracture of the skull that tore the middle meningeal artery and caused an internal cranial bleed that eventually caused Goliath's death. Okay? So if you're really good with a slingshot, it is a dangerous weapon. Tell my grandson that he is. If it's dangerous, he just goes right for it. but this is the middle meningeal artery and the posterior meningeal artery. So this is right over, you know, coming right out adjacent to the squamosal portion of the temporal lobe, okay, as it's coming up here. All right, if we look at these three surfaces and it's kind of a cutout, we see them laid out by the author here. Here's the cerebral cortex, okay? And these are all neuron populations in here. This is the dura mater. This is now the periosteal side. There's a subtle space between the dura mater and the arachnoid mater, and here we see it coming down into this one cell layer thick area, the pia mater. And these are the arachnoid trabeculae, okay? In this case, it's covered, they're showing covering over an artery. But if you notice here, you see the pia mater following these arteries deep into the brain. This is how pia mater becomes part of this area, this protective mechanism we call the blood-brain barrier. It helps to prevent large molecules from coming in contact with neurons. Okay? So we see the pia mater really following the arteries all the way in. Okay? This is the subarachnoid space. There are also venous channels in here that is not really showing. This is the subdural space. Now you've heard about, you know, probably over the course, I don't know, Ashley, you do soccer coaching. Anybody else do any type of athletics, like, you know, lacrosse or even baseball or things like that? Okay. You ever have an athlete get a epidural or a subdural hematoma, where they had to be transported to the hospital? Okay. So there's a significance about that because the consequences of an epidural hematoma take place a lot slower. Why do you think that is? Why do you think that is? An epidural hematoma. So it's now outside the dura. So that's a pretty tight layer. So if it's bleeding in there, that tissue is tight enough that it's going to take a lot of pressure over a long period of time to really have an internal effect. Okay? We'll talk about the consequences of why. But if it causes a laceration of the endosteal dura, it will bleed into the subarachnoid space. Just like, for example, if there is a venous rupture on the surface of the brain, that'll bleed into the subarachnoid space. Or if there is a blowout of even small arteries on the surface of the brain, they will also bleed into the subarachnoid space. And that will take place a lot faster. So we've got these epidural hematomas. So this is the subdural space. So if there was bleeding in here, it would now be compressing the subarachnoid space. Out here, it would be tearing away the dura from the inside of the skull. Some of you may at some time continue your coaching careers or things like that. A lot of dentists, a lot of podiatrists I know do that. They work as sports physician. You maybe get lucky enough and be selected to be the dentist, you know, for, if you're lucky, you should get the Golden State Warriors because they're more consistent, you know, but maybe the Cavaliers. Pretty much, yeah, pretty much. Okay, so what happens to the dura? Because the dura is growing at the same time the brain is growing. And the brain has to have some support, okay, for a lot of reasons. One, the cerebral cortex lives up here. Here's one side, okay? So here, for example, you can kind of see this. This would be a temporal lobe. We'll talk about the brain, the lobes of the brain in a few minutes. Temporal lobe would be lying here. Here, the occipital lobe back here. Here would be the frontal lobe and the parietal lobe up here, lying against here. But this wall, this falx, remember we talked about what the term falx means? Okay, remember we had the conjoint tendon? The conjoint tendon was a falx which created a wall for the inguinal canal, in particular in males. Okay? This is called the falx cerebri. This separates the left cerebral cortical hemisphere from the right cerebral cortical hemisphere. What's lying in here would be an area we refer to in the brain as the diencephalon, the thalamus, the hypothalamus, not basal nuclei, because basal nuclei are actually part of the upper portion called helencephalon, but also the beginning of the brain stem, the midbrain, and then coming down the pons. What would sit in here? Well, if you could read that. The cerebellum would be sitting in here. So we've got this dural fold that actually creates a roof over the cerebellum, so this real large cerebral cortex doesn't wind up chronically putting downward pressure on the cerebellum and causing issues with things like coordination of movement. Okay? So we have the cerebellum moving down here. Okay, this down here, this is just a maxillary sinus. Here's your orbit, maxillary sinus below. Okay, here's this is probably the frontal sinus up here. So we see these folds, these dural folds, that help to provide support for primarily the cerebral cortex, the cerebellum, and upper portions of the brainstem as it descends down towards the spinal cord. So the other thing that the dura mater does is it creates venous channels, and that one venous channel we talked about on the very top, between the dural layers, is this superior sagittal. Okay, this line running down the midline of the body is called a sagittal plane, okay? Dividing the body anywhere into a left and a right. This is the superior sagittal sinus. Actually, remember we talked about a foramen right here? What was that little foramen in front of this crest called? Foramen cecum, right? Where did that communicate? Yeah. What if you had a severe nasal infection, nasal cavity infection? That could get up into here. Okay? So those are things that you always have to remember, okay? So, and then down below it is this falx cerebri, and then there's another very, very thin. We'll look at, I think, we'll probably be able to get at least one brain out where we can see the folds very clearly. But we do have a couple of brains that are already out of the cranial vault. But this one is down below. This is a little bit deeper. So this is the inferior sagittal sinus. Both the superior sagittal sinus and the inferior sagittal sinus, we're going to see, has an interconnection with deep cerebral cortical venous channels. And that's how the venous drainage of the brain gets back into circulation. So we see here, superior sagittal sinus.
Dr. Hernandez, what are the sagittal sinuses interconnected with?
What are they?
With what are they interconnected?
They're eventually going to be interconnected with the jugular venous system draining the cranial vault. Okay. Then we have the inferior. Okay, and then coming off of the inferior, running over the midline of the top of the cerebellum is this venous channel. It's just a straight vein, so they call it the straight sinus. Coming off in the back of the superior sagittal sinus here, most of the time, we talked about this in the lab on Saturday, most of the time it'll drain over into this right sinus that's running transversely from the superior sagittal sinus out to the right side of the skull, and it creates a groove there. And this is called the right, bless you, transverse sinus. And typically, the straight sinus drains into the left transverse sinus. So let's do this. Okay, so we'll see, we'll go into, look at the deep veins in just a minute or two. So we then have, once again, there's forum and cecum. Okay. What's the sinus embedded in the lateral wall of the sphenoid bone deep to this region, the cellulose? What's that called? Cavernous sinus. Good. Okay, and what vein is coming out of the orbit to drain into it through the superior orbital fissure? What vein comes out of the orbit? What region do we call the orbit as a region? What's it called?
Optic?
Optic or ophthalmic? Okay. So this is the ophthalmic vein. Is there any vein from the face that drains into the ophthalmic vein?
First.
Which one?
Isn't the first the trigeminal?
Is it trigeminal venous channel?
Oh, no, I'm sorry. Isn't the first one though?
Okay. What venous channels drain blood from the face? Like facial vein? Okay. Do these veins have valves? No. So the facial vein, actually the facial artery as well, actually have interconnections with vessels in the orbit. The venous channel can then bring bacteria build up in the face, and that bacteria can get back here into the cavernous sinus and cause a relationship where we get a venous channel inflammation called a thrombosis, and that can cause problems with the, maybe the young adult comes in with a fever of unknown origin, okay, and is very ill, but he has a very high fever of somewhere around 90, about 100, 105, 39 degrees centigrade, or something like that, okay, with bacteria draining now through the orbit into the cranial vault. It's a very common infection. It's called a cavernous sinus thrombosis. Then from the cavernous sinus, what's this portion of the temple bone called? Yes.
I don't mean to be stupid, but I'm not following you. So bacteria comes in through the nose or the mouth, and then it gets
Nose of the
this venous
face.
system somehow?
What's that?
It gets into this venous system somehow?
Oh, yeah?
I'm not following that. What's the chain?
Okay. The thing is that all the veins are connected. There's no valves to prevent any type of blockage of venous flow. The thing that modifies venous flow is gravity. Okay?
Oh, as you're coming into the turbinates, you've got to get from those to the veins, yeah?
Oh, yeah.
Are they crawling through, or?
Okay, how would veins from, let's say, around the nasal cavity, even the infratemporal fossa where the cranial nerve 3 lives, or something like that, how might they get into the cranial wall? Are there any openings?
Well, there are sinuses, but are those highly vascularized, or?
Sinus is just an air, we're talking about air sinuses now, maxillary frontal, okay? But we have venous veins, we have veins. We have veins that, for example, are called the sphenopalatine vein, okay? Which drains into a venous plexus called the pterygoid venous plexus. But it also interconnects the posterior pharynx adjacent to the underside of the sphenoid bone, okay? With vessels adjacent to the sphenoid bone like the cavernous sinus. There was this, what I showed you an opening in one of the skulls that isn't always there, isn't always easy to see. It's one of the hardest ones, someone doesn't see. It's in front of foramen ovale, what do they call it? It's described by an ancient, Greek ancient, Vesalius, yes, okay, foramen of Vesalius. What if there are no foramen of Vesalius? Where might it drain? How might it get into the skull? Foramen ovale, okay. How else? And this is where Landers differentiates with some other anatomies. Foramen lacerum. Okay. So all of these veins are interconnected. What are, Brian, what are veins draining?
Any area that's perfused, I suppose.
Right. Any area that's perfused. Can bacteria then from an infected area get into the venous channel? Yeah. Okay. Get into lymphatics as well. Okay. But if it gets into the venous channel, it's more dangerous because the venous channels can go deep intracranially or drain deep intracranially, as opposed to, let's say, back through the facial vein into the external jugular or internal jugular vein. But that's dangerous enough because that can bring bacteria into the entire circulation, systemic circulation. Okay? What did I tell you about infections? Is there such a thing as a simple infection? No. Means the body has basically, you've got this micro course going. I know you're focusing on complement and things like that now, immunology, but bacteria don't care how they get there as long as they get there, okay? Do bacteria eat tissues? No. But they live off of them. They live off of the same things that the tissues are using for their metabolism, okay? So, a lot of times we're lucky, okay? We win, the bacteria lose. Sometimes we're not lucky. The bacteria win, okay? So, that's why you have to understand this. You have to know this, because you are dealing with probably an area of the body that might be considered a sewer. That's the oral cavity. What type of bacteria live on the skin?
Aerobic?
Staph. Okay. Staph's a rather virulent bacteria. Have you talked about that? Have you talked about vectors? No? Okay. So you don't want staph into deep tissue spaces. Okay. And you definitely don't want staph intracranially. How does venous flow from the dentition drain? Into what tributaries? In the mandible, where does it drain into? What's that called? What's that called? What's that branch called? You've got the arteries called the inferior alveolar. What's the vein called? Inferior alveolar. Okay, in the maxilla, you've got essentially three primarily drainage regions. Okay, you've got the anterior superior region, you've got possibly a middle superior region, which actually those two run through the maxillary sinus. And you've got the posterior superior region. Where do they drain? They drain into this infratemporal pterygoid venous plexus area, which has intercommunications between the foramen vesseli and the cranial vault. Okay? Teeth can kill you. In many cases, over the course of history, they have. What?
Yeah, so you have some periodontal disease, there's been significant bone loss. The bacteria would go from the area bone loss to the veins and then into the brain, or into the jugular, into the heart?
Depends upon how the gravitational forces are, whether you're lying back, whether you're standing on your head,
Okay.
okay? Things like that. So that's something that you always have, to think about, in relationship to that. So, you know, when patients used to say to me, what's the big deal about it? What's the big deal, doc? This tooth decay can't kill me. I said, well, not yet. But if you want to let it go a while, maybe it will. Okay? But you have to know that. They don't. I got news for you. Physicians don't even know. When somebody comes in with a cavernous sinus thrombosis, they're thinking if they got this high fever, they got some type of pharyngitis. Then they look in the pharynx, there's no inflammation. Then they start looking at other places, like they look at the cheek area, the deep cheek area, and they see that it's a little swollen, and through the acne, and the acne is all inflamed, and there's drainage there. Then they say, okay, maybe we need to look at this a little closer and put this patient on an antibiotic drip. Okay. And cavernous sinus thrombosis still happens in this population in the Western world. So it's something you have to think about. I have patients that, for whatever reason, wound up with brain abscesses in relationship to dental or periodontal infections. Okay. And fortunately, they were picked up before they killed the patient. That's your job. Okay, so the cavernous sinus can also drain back here. So nobody answered what that bone was, where the X is. Deepest portion of the temporal bone, okay? So there is a thin groove. If you look at a temporal bone, or you look in the skull, there's a thin groove on the top. And that groove is formed by a dural sinus, and this is called the superior petrosal. Okay. Coming down deep from the cavernous sinus, we'll have another sinus that'll come down here, and this is referred to as the inferior petrosal sinus. One side. Okay. All right, so now these transverse sinuses come a little bit forward along in a horizontal plane, and they will make a S-shaped turn as they come down to join what vessel that's draining the skull? What's the venous vessel that drains the skull? Drains the cranial vault from the jugular foramen. Internal jugular vein. Okay? So that's what's down here. What would you call a venous channel that's shaped like an S? Sigmoid. Okay. So this here is the sigmoid sinus. So with the petrosal sinuses, the superior petrosal sinus drains into the junction of the transverse sinus of the sigmoid and the inferior petrosal at the junction of the sigmoid and the jugular vein down here. Once again, there's no valves here. If anything's going on down here, down below, and for whatever reason the bacteria, due to gravitational forces, start to move upstream, they're going to go inside the cranial vault. Okay. They go inside the cranial vault.
And where did the superior petrosal drain into? You said the inferior petrosal drained into the junction.
Superior petrosal drains into the junction of the transverse and the sigmoid.
Okay.
The inferior petrosal drains into the junction of the internal jugular vein where the sigmoid sinus meets it.
Okay. Thank you.
Okay, so here's kind of a transparency that shows the same thing. Okay? We've got this network in here. These are spinal and brainstem veins. There's your superior petrosal. Your superior petrosal. Okay, here's your inferior petrosal. And here's your sigmoid. All right, so the dura is a very dense connective tissue membrane that's providing structural support for this large object inside of this bony skull called your brain. But along with that, what's forming in here are a vascular drainage mechanism from the brain itself back into circulation, and those are called dural venous sinuses. Their inner lining does not represent an endothelial lining similar to what you see in a true vein, but there is a inner lining. Okay, so they are venous tributaries or venous channels, but they are not true veins. They're just Here we see the arachnoid. Over here, okay. What do you think this is? Alongside the cerebellum. That's a transverse sinus. This is the left side of the brain. So this is the left transverse sinus. Here's the cerebellum. Okay. Cerebral cortex is covered with arachnoid. Here you can actually see some of the, we'll talk about these in a few minutes, the arterial channels coming up here. They're primarily branches of the middle cerebral artery. These are internal carotid branches. They are also covered with arachnoid.
Is the falx cerebelli still attached in this picture? Is the falx cerebelli still attached in this picture?
Oh, I'm sure it is.
Okay.
But you can't see it because you don't have a complete separation of cerebellar hemispheres.
Okay.
Okay? But you do in the back of the cerebellum as the some hemispheres kind of go laterally a little bit. You do have this indentation created by this dural wall, this thin, small dural wall called the falx cerebellum. Okay, you want to take a break? So there is, just for your reference, there's some things online that are worth getting if you want. One of them is called Complete Anatomy. It's a three-dimensional atlas. You can move the images around. Okay, I think I've got it. I think it's as good, if not better, than the whole ones. Okay. And we're working on that. We got it, next week we're taking your GI, and then we're gonna try and set up those of you who signed up to look at the whole graphic stuff with the GI demonstration. Okay? Then there's another one that is called, it's called 3D Anatomy for Artists. It's done by artists, which is actually also really good. So both of these I know are iPad or I don't know if you could probably do it, download it to an iPod, but an iPhone. I'm not sure if they are, you know, like, Android capable. I would hope that they are, but I'm not sure. Okay, but they're definitely
iOS cable, and they're really big. They're actually using, they started using it for medical schools, both the 3D anatomy for artists and the complete anatomy. Okay. Also, I got to see, I don't know if you've seen this, they've got this, what's it called? Not Avacore. That are going to be the demonstration devices used over in the HEC. Basically, it is a computer with an 85-inch screen that runs Microsoft. Okay, so there are better things than that, but they'll work that out. Okay, that's a lot of the demonstrations, in particular in the small rooms. Those are probably going to be available in all the small rooms and in some of the larger lecture rooms. Okay, wherever those may be. They haven't told us yet, so.
They've got my graduate course this summer, and I really don't want to be over there this summer. So, with the medical school building, they're trying to redo the anatomy labs because they're still going to be cadaveric anatomy. Those are things that are coming around. Okay. So, we're going to kind of take an overview of this structure because we're going to see it in the lab. Actually, this is the only place we're going to see it is in the head and neck lab. Okay, we're not going to we do not have a neuro lab. So the only place we're going to see the brain is in the head and neck lab. So we've got this large bulbous structure called the cerebral cortex. Okay, shell. The cerebral shell that is surrounding all of these neurons, okay, and giving them a place to live, okay? And there are 5 lobes of the cerebral cortex per se. Some people would say that there are 6. We're not going to talk about that now, we'll talk about it in the neuro. And that's because there are regions that are called the homotypical cortex, or the cortex of man, or the neocortex, okay, the new cortex. And then there are areas that are the oldest areas of the cerebral cortex, which are called the archicortex or the allocortex, or some regions of those are referred to as the limbic system, the limbic cortex, or the insula. Okay? So the insula kind of falls in over two regions here. So we've got a frontal lobe, parietal lobe, an occipital lobe, a temporal lobe, and an insula. They say that in the central nervous system by itself there are 100 billion neurons. You know, this always amazes me. I said, so show me the calculation, you know. And it's usually the same as they would do for things like a blood smear. They would take an estimate of a region, they would take the area of the cortex, and then they would multiply it by a constant. So 100 billion neurons. They say we only use 10%. Do you believe that? I do. And why do you think that is? The ones that we use, that there's only 10%.
I'm not really sure where that came from. You have to use 100% of your brain. If you get a stroke anywhere in it, it's a problem.
Well, that's
There's
true,
not some vast
but
90%
what
that's never used.
are the mass, what's the largest amount of neurons used for? To store information, memory. Okay, it's what we call association cortex. We'll talk about that in a minute. So, but the things we use that we receive sensory input or we put out motor output, those are the 10% that are telling all of the other organ systems in the body what to do for receiving information from those organ systems. But the majority of these cell populations is your memory, okay? And you can believe what you want, but there are, you know, it's kind of, most neuroscientists believe that we are a 6 million year old organism from start to where we are now, okay? And that over the course of 6 million years, our brain has grown because as we have lived in these environments where you are predecessors have lived in this environment, they have gained information or gained information to use to get you around in that environment. So we've actually, like the brain is grown like a flower off of the brainstem. Kind of interesting. Okay, so a couple of principal areas we have. We have a fissure here, or a sulcus. These ridges are called gyri. And these grooves between them are typically called sulci. And this one in particular is kind of important because this is called the central sulcus. The full name of Rolando. Rolando Gattara. That's his heir to fame. And so that central sulcus divides this lobe here. This is the frontal lobe. Let's do a different color. That's not working. From this lobe here, which is the parietal lobe. Then there's a little notch, just kind of got moved off here, okay? And this is called the parieto-occipital notch. It's actually bigger on the medial side, we'll see. That separates the parietal lobe from this area here, and this is the occipital lobe. Okay, so that's three of the 5 lobes. And then we have this large fissure in here, because it's very deep. This is called the longitudinal or Sylvian fissure. And that separates the frontal and the parietal lobe. It's in front of the occipital lobe. This is the temporal lobe. So that's basically 4 of the 5 what we consider to be true cerebrocortical lobes. Okay? Here is a schematic. This is out of a book by Dwayne Haynes. Dwayne Haynes wrote a book called Neuroscience Basic and Clinical. That's pretty much the neuroscience text we're going to use, and it's pretty well distributed throughout the country right now, medical schools, dental schools, nursing schools. Okay, so here's frontal. Here is the central sulcus. Here's parietal. Here is occipital. And here's temporal. We turn the brain, we split the brain in half and look at the inside. Did you have a question?
Where's the longitudinal sulcus on here?
Right here.
Is longitudinal same as lateral?
Yeah. Yeah. Longitudinal laterals or Sylvian fissure. Okay. Here's the medial side. So we have the superior fissure comes over the top. Okay. Here we've got, here's our brainstem. Over here with the midbrain and the pons, and then coming down to the medulla. This is a commissural tract coming from left to right or right to left. This is called the corpus callosum. Over here. And coming over in here, this is the central sulcus. So this is parietal back here, occipital, frontal over here. Let's get a good view of the temporal on this from this perspective over here. Okay. So this is a intersection between the parietal lobe and the frontal lobe called the paracentral lobule. Talk about this in a minute. Okay. Here we see, here's the cerebellum. And scientists thought that it looked, when it looked in this midline cut, the sagittal cut, that it looked like a tree with branches. That the cells, the neurons, were the leaves. And the branches were the white matter that was coming from the leaves to the core. So here are the leaves out here. Here are all the neuron populations, and here are the branches. So they said this was a tree. And they came up with the term arbor vitae, tree of life. I mean, if you lose a cerebellum, is it going to kill you? No, but you're probably not going to do very well in your environment, okay? Because it is primarily the center of motor coordination. All right, here is a schematic. Here, once again, frontal, parietal, occipital, and here's a little bit of the temporal lobe. The fifth lobe is kind of hidden deep to the temporal lobe in this lateral, longitudinal, or sylvian fissure. So we have to kind of pull it back. And that is referred to as the insula. And this is the archicortex. This is the oldest portion of the cortex. All of this out here is the neocortex. And you can see just by looking at it that the diameter or the of these gyri, of these folds here, looks a little bit wider, thicker than the gyri here. So why is that? Why is that? What's the difference here and here? Okay. This is the neocortex. The neocortical gyri are, for the most part, from outside in, are 6 layers of neuron populations we'll talk about in neural, okay? From layer 1 all the way down to layer 6, okay? So here, though, at most, maybe you will get 5 layers, but it's usually somewhere between 3 or 4 layers of cells. So not as much developed there. This is part of a system we call a limbic system. What does the limbic system do? The limbic system kind of oversees our emotion, our behavior, okay? Just the basic things we need to survive in our environment. And initially, that's what humans needed. They needed a baseline to survive in their environment. Okay, all right, so let's go deep inside of the brain, so to speak, and look at some of these deep venous channels, okay, that we don't really see. We see some of their extent on the outside. They interconnect the dural sinuses to the venous channels on the brain. So the intricacy of this is really from two fairly large venous channels interconnecting the largest sinuses. And so the largest, longest sinus is the superior sagittal sinus, and it's interconnected by an anterior superior vein that was described by a neuroscientist named Trillard. And this anastomoses with the superior sagittal sinus and with some of the posterior inferior veins, a vein referred to as the vein of Lavi, which then anastomoses with the transverse sinus. So these are veins on the surface of the cortex itself that are actually interconnecting venous flow from the superior sagittal sinus or from the transverse sinus through these venous return mechanisms on the surface of the cerebral cortex. Okay? So the vein of Trillard to the vein of Lavi, we'll talk about
in greater detail. Deep inside the brain, there's some other venous channels that are associated. This deep basilar vein of Rosenthal kind of runs on the inside of the temporal lobe, okay? And on the top is the inferior sagittal sinus. So there's kind of a venous channel here and a venous channel here. And they join a really short little vein that drains, we'll see in these two images, drains into this junction with the straight sinus running over the top of the cerebellum. Okay? So let's look at some of these. Okay, so here's the anterior anastomotic vein of Trolade, and here is this vein of Labe. I wish I knew why it does this, but I'm starting to get a kick. But this is the vein of Labe. This is the superior anastomotic vein of Trublar. Okay. This is the vein of Labe. Here's a transverse sinus. Here's a superior sagittal. We don't see the inferior sagittal because that's deeper inside the brain, but once again, if we do this sagittal split of the cerebral cortex, we now, here's superior sagittal. Here is the inferior sagittal. Here's the temporal lobe. This is the vein of Rosenthal. And this is this vein of Galen. And this is the straight sinus. So what are these veins doing? Well, they're taking the venous return from the neuron populations of the brain itself, okay? And they're taking that venous return into these dural sinuses to go into the systemic venous circulation of the head and back into the systemic circulation of the body. Yes.
What's different from the view from the slide before and then this one?
We're looking at the medial side of the cerebral cortex. The slide before, we're looking at the lateral side. Okay. So this venous return, what percentage does the brain have as a metabolic organ in the body? What percentage of the overall metabolism does the brain have? About 25 to 30%. It receives about 25 to 30% of the output of the heart, and 25 to 30%, therefore, of the oxygen being carried in the brain, carried to the brain from the circulation of the heart. So it must be pretty important if just this area in here is has this metabolic activity in order for us to basically think and do things. Right, here's kind of an overview of these venous dural sinuses. Okay, once again. Okay. These are these little one-way valves we'll talk about. These are called arachnoid granulations. Okay,
And here, Brian, this is for you. These are all of the venous tributaries from the intercranial portion interconnecting with venous tributaries of the head and the neck. There's no valve between the highest venous tributary and until we get down probably to the junction on each side of the internal jugular vein with the subclavian vein forming these brachiocephalic veins on each side of the neck. Okay? No valve. So blood flow is determined by gravitational forces. Okay? So anything in that blood will flow with it. All right, let's look at arterial blood supply. Okay? So we've got two large feeder areas that are dealing with arterial blood to the brain. You know what they are? Well, you can imagine. Okay, what's one of the large arterial blood supplies to the head and neck? What is it? The carotids. Okay, the common carotids. Okay, the left common carotid comes off of where?
Aortic arch.
The aortic arch. Right comes off of brachiocephalic. Why is it called brachiocephalic?
It's a connection of the arm and the head.
Arm and the head. Brachium is the arm, cephalic is the head. Some people call that vessel the humiliant. As the arch comes over, that's the first branch that's given off to the right, okay, side of the thorax. So then the carotids come up about the level of the thyroid cartilage, this cartilage on the outside of your voice box, so to speak, your larynx. And it branches into two branches, one more anterior than the other, and one with branches externally, but the other without branches externally. So we have right and left internal carotid arteries, which are going to not branch, and they are going to go up and enter into the skull to supply the structures within the confines of the skull, including the retinal surface, the ophthalmic artery, the orbit itself, okay? Also, all of the areas of the cerebral cortical hemispheres, some areas inside the internal ear we'll talk about as well. So there are three branches that are given off the right and left internal carotids, and you'll see those in the schematic. The anterior cerebral, the middle cerebral, and there's a communicating artery, a tiny little artery that joins the left and right anterior cerebral artery. The posterior branches come from the left and right vertebral arteries. Where do the vertebral arteries branch? Where do they branch from? This is why now you have to start studying the words. Okay. Where do the vertebral arteries branch from? What larger vessel? Subclavians. Subclavian artery on each side gives off a vertebra. Which branch of the subclavian does the vertebral artery represent?
Is it the inferior?
It is the first superior branch of the subclavian. What's the first inferior branch of the subclavian? If you're feeling stressed now, wait until you're taking an examination. Okay? Okay. You looked at the stuff in the thorax, right? You look at that stuff, what do we talk about? What were these two arteries running parallel to the sternum, deep to the thoracic? What were they called? Internal thoracic or internal mammary? That's the first inferior branch of the subclavian.
inferior branch vertebral, inferior branch is the internal thoracic or internal mammary, okay? That will come in through what large opening? If you were in a lab session on Saturday, we talked about that. What's that large opening in the bottom of the skull? More magnet, okay? So they come in there. How did the internal carotid come in? How does that come in? What does that mean?
Carotid canal.
It enters into the carotid canal for forming it's got a kind of a confusing name called the external carotid foramen. Okay? So this comes in and gives spinal arteries off. It gives off a artery on the underside of the cerebellum. It gives off an artery that runs on the underside of the pons, that's the base of the pons. So it's called a basilar artery, which gives off anterior, inferior, and superior cerebellum. And then the last branch of this basilar is typically the posterior cerebral artery. And then between the posterior cerebral artery and actually this artery here, okay, and as it's given off from the internal carotid, is a posterior communicating. So this forms what's called a vascular circle. And it was described by an anatomist named Willis. So it's called the cerebral vascular circle of Willis. So the blood supply to the brain about, only 2% of the body weight, but anywhere between 20 and 30% of the metabolic output, okay? 20% of the inspired oxygen is used by the brain. It's basically taking 20% of the cardiac output. Okay. So if we were using, Brian, 100 billion neurons at the same time, there would be smoke coming out of our ears with that much energy being used. Okay. All right, here are these vessels. Okay, here is over here. So this is the aorta. This is the subclavian on the left side, all coming off the aorta. Here is the left common carotid coming off. Where it's branching, where this vessel splits here, around the thyroid cartilage, where it splits or bifurcates, there's a little enlargement called the carotid bulge or the carotid sinus, which is more from a physiological perspective. What was going on there? We talked about blood pressure. Baroreceptors. What else? What else? Chemoreceptors. Okay. The chemoreceptors are inside. The baroreceptors are in the wall. What are the baroreceptors monitoring? Blood pressure. What are the chemoreceptors monitoring? Okay, and what does CO2 in solution create? Acid. It's monitoring pH. Okay, so that's what's going on in here. So this comes up and splits. The branch that's more anterior that has all of the sub-branches to the neck and the head, that's the external. The branch that comes off that has no branches until it enters into the skull, that's the internal. So the internal carotid does not branch until it gets into the skull. The external branches almost immediately once it splits off of the carotid sinus. Okay. Here's down here. This is, I think this is also, because this comes out of a tumor book, I think this is also in Eric Baker's edited head neck dentistry or dentistry for dentistry. Anatomy for dentistry. So this is a internal carotid coming through the carotid canal. Do you think that this is a problem when it's first coming in here? Do you think that that is a problem?
I
Okay.
don't understand the question.
The way that this bends. Do you think that's a problem?
I guess just however it's gotta get in there.
Okay. Do you think that if blood is going through there and possibly atherosclerotic plaques are building up in there, They think that that could possibly cause a blockage of the entire arterial flow.
So here is the front of the brain area. Here are the internal carotids. Going out laterally is this vessel. And this is the middle cerebral. Going over here anteriorly, this is an anterior cerebral. And there's an anterior cerebral on each side, and between them, joining them, is this tiny little vessel here, and that's called the anterior communicating. Okay, then here are the vertebrals back here. This is kind of cut off. There's two vessels here. The one that we're seeing here, this is the anterior inferior cerebellar. It got cut off here. Is a vessel that comes off almost immediately as the vertebral enters into the cranial vault, called the posterior inferior cerebellar. Then we have this vessel here. This is the basilar. And then the basilar will give off a couple of branches up here. This one is called the superior cerebellar. And this one is called the posterior cerebral. So then there is a vessel that can join the anterior branching with the posterior branching. You see that here? That's called the posterior communicating. So if all of these vessels are present, what do we have? We've not only got cerebral blood flow, but we've got a cerebral vascular circle that actually has a relationship bilaterally to the left and right portions of the cerebral cortex. Okay? Now, there's usually always an anterior communicating, but there's not always a posterior communicating. So that can actually create a situation that makes that individual more labile to possible things like stroke, cerebral vascular accidents, or some type of issue where there's a blockage to one side of the cerebral cortex. If we look at the lateral side of the cerebral cortex here, this is the middle cerebral. So it's coming through here. All of these branches are going frontal lobe, parietal lobe, temporal lobe. Okay. This back here is the posterior cerebral. These are cerebellar. we once again cut the brain in half and look at the medial side. Here is the anterior cerebral. And then this small section back here. This is the posterior cerebral. So if the middle cerebral is such an issue because it takes care of about 2/3 of the surface of the brain, of the lateral surface of the brain. It takes care of the frontal lobe, the parietal lobe, the temporal lobe, okay? It's associated a lot with deficits caused by blockage or bleeds, with language issues, being able to express language or understand language, okay, in those areas. Here's the vertebral artery coming up. Here is the posterior inferior and posterior superior. Okay, so those are coming up. Yes, Frank?
Okay. Where is the insula found versus the corpus callosum?
Okay, the corpus callosum is here.
Yeah, that whole. Just the white part or that whole region?
Just the white part. Okay. The insula would be deep in here. Okay.
So does
It's
the
covered up by the temporal lobe.
Does the corpus callosum surround it or no?
The corpus callosum is what we call a commissural pathway. Neuro will talk about commissural projection and association pathways. Commissures take information from left to right or right to left. So an interconnection of the right side of the brain to the left side of the brain. This is the largest commissure. So right cerebral cortical hemisphere with the left cerebral cortical hemisphere. Okay. Projection fibers take information either from lower centers up to the cerebral cortex, or from the cerebral cortex down to lower centers, including the brainstem and the spine. They project to or from. Association fibers project either from the front to the back, or from the back to the front. These are really more discussion we'll talk about in greater detail in neuro. Okay.
Does the insula communicate with the medulla? Or the,
Here, I'll show you some. Okay. This gyrus here is called the cingulate gyrus. This actually intercommunicates, comes down here into the temporal lobe via a gyrus that's called parahippocampal. With this structure here, which is referred to as the uncus. So those are part of the limbic cortex. The cingulate gyrus interconnects the neocortex, okay, the homotypical neocortex, with the archicortex, the insula and associated structures such as hypothalamus, for example, but with the limbic cortex. Sometimes the cingulate gyrus is called the limbic cortex. Okay. So these are the main large vessels. So we'll be looking at the gross brain. We're not going to be talking a lot about, other than some of these large vascular structures and some of these regional questions, so much as we're going to be talking about them next semester in neuro. okay, and what the consequences of things going on in them if something blocks or something bleeds. So there are tiny little cerebral branches that come off of the posterior cerebral artery and the anterior cerebral artery that actually perforate the cerebral cortex and go into its chambers, its spaces, that are called ventricles. And within the ventricles is a substance produced from the filtration of blood called cerebrospinal fluid. And these vessels that come in are referred to as either anterior choroidal or posterior choroidal vessels that interconnect from a histologic perspective. They a very tortuous, membranous-like sieve, okay? And this is called the choroid plexus. Okay? There's a choroid plexus that's typically we'll see in the junction of the two of the ventricles, actually three of the ventricles, and one that is sitting underneath the cerebellum in one of the ventricles in the back. So cerebrospinal fluid is really produced more anteriorly into two very large, and placed into two very large ventricles that are called lateral ventricles. And they are separated each other by a membrane that is down below that corpus callosum that we're looking at, called the transparent membrane, the septum pellucidum. Okay? And so there's a chamber on each side of that septum pellucidum. It's called a lateral ventricle. If we look at it, these ventricles in a certain plane look like ram swans. They look like large ram swans embedded inside the brain. From there are two little openings from each side, from each lateral ventricle that flow the cerebrospinal fluid produced in the lateral ventricles primarily, down into a ventricle between this sensory relay nuclei, this bilateral sensory relay nuclei, in an area we call it diencephalon. But the space between the thalamic hemispheres, the one on the right and the one on the left, is called the third ventricle. So from the lateral ventricles, the cerebrospinal fluid flows down, once again gravitational forces, from the lateral ventricles into the third ventricle. So those are called interventricular foramen, and they were described by a neuroscientist named Monroe. So they are called the interventricular foramen of Monroe. Then from the third ventricle, there's a thin little tube that travels down the center of what we refer to as the midbrain, the upper portion of the brain stem. Okay? And on the back of it are four large bumps called colliculi, or together they call them the four bumps, or the corpora quadrigemini. Okay? And underneath them is the base of the midbrain. And that was described by Sylvius, the same guy that talked about the lateral fissure, or the longitudinal fissure. And this is now called the aqueduct of Sylvius, or the interventricular aqueduct. Okay? And this is called this is the junction between the third ventricle and the fourth ventricle. The fourth ventricle lies between the back of the pons, the posterior surface of the pons portion of the brainstem, and in front of or under the cerebellum. So there's also a choroid plexus in there that's also producing cerebrospinal fluid. Okay, so where does the cerebrospinal fluid have to go next? It has to go out and flow into this subarachnoid space, which is outside of these ventricular chambers of the brain, and surround the brain with this fluid. And so there are some foramen in the back of and on the sides of the fourth ventricle that were described by Lucia and Magindi. The ones on the side are called foramen or foramina lucia. The ones in the back is called a foramen Magindi. Okay? And those foramen take the cerebrospinal fluid from the ventricles into the subarachnoid space. So here, looking at the front of the brain here is the lateral ventricles. Okay, here are the foramen of Monroe, or the interventricular foramen. Here's the third ventricle. Here is the cerebral aqueduct of Sylvius, going from the third ventricle into this ventricle here. This is the fourth ventricle. And this is the cerebral aqueduct. And then these openings here are the foramen of Luschka. And this foramen here is the foramen of Magenta. Okay, there's also another foramen that doesn't go into the subarachnoid space. It goes in a canal that runs right down the center of the spinal cord, called the spinal canal or the central canal. Here's another look. Lateral, third, interventricular foramen. See how large this is, the lateral ventricle? Here is the aqueduct. Here is the fourth ventricle. Okay. Now, the cerebral spinal fluid then goes out into the subarachnoid space. And subarachnoid space is not just surrounding the brain, it's surrounding the spinal cord. It's surrounding the spinal cord. And this is an ultrafiltrated blood, so what are we carrying to cells in blood? The nutrients they need to live. Okay? And we're returning the waste from their metabolism back into circulation. So this is a choroid plexus in the lateral third ventricle border. Here is a choroid plexus in the area of the fourth ventricle here. So we're producing the cerebrospinal fluid here. It's flowing down here, it's flowing down here. Then this out here would be a foramen of Magendae, and coming out to the sides would be foramen of Luschka, and they would be distributing that cerebrospinal fluid all the way down surrounding the spinal cord. Okay, so now it's going to circulate. It's going to go all the way down and around and come all the way back up. And here's the subarachnoid space. This red line is the pia mater. This is the subarachnoid space. This is what sinus? Superior sagittal sinus. These are one-way valves called arachnoid granulations that return the cerebrospinal fluid, just like other venous channels, into the venous channels to go back into circulation. The superior sagittal sinus is now going to take this ultrafiltrate and the waste products in it back into circulation. Cerebrospinal fluid, they say, has a pressure somewhere around 30 to 50 millimeters of mercury pressure. Okay? That's why when this is starting to form, you see in this membrane bone, in the calaveria, you see these little fossa, these little depressions called pachyonian depressions. You know, pachyderm is a large animal. These are large depressions. So it comes back into the superior sagittal sinus. That's how cerebrospinal fluid is returned. How is it getting there? It's getting there through these arachnoid granulations, which are draining cerebrospinal fluid
from the subarachnoid space. Here's the arachnoid mater, here's the pia mater, here are the neuron populations here. Here down here, this is like this is one of the cerebral cortical branches here. Here is this falx cerebri here. Here is the superior sagittal sinus, and that is receiving not only the drainage of cerebrospinal fluid back into circulation, but it's also receiving venous blood from the surface of the brain, okay? It's also draining in here, or possibly surface of the dura. Okay? So that's it. We'll talk about these again next semester. We'll talk about some of the more clinical related things that happen when things in here kind of go haywire. We may see, I mean, sometimes we will see when we're taking a brain out, we may see an individual with severe brain damage. Brain, it's interesting to see, but it doesn't do too much for us as far as looking at the brain. Or we may see a tumor. Okay? Things like that. One of the problems these days from an anatomical perspective is people and their families don't like to do autopsies. So it's just an education.